And before we move into microRNA, I'll just touch upon glaucoma and where microRNA actually fits into this. Most of you may be aware that glaucoma is a group of eye diseases and the most common cause of irreversible blindness worldwide, and mainly characterized by etiologies that can lead to elevated intraocular pressure, retinal ganglion cell degeneration, and optic nerve damage, which, if non-treated, eventually leads to loss of vision. So why glaucoma? So this pie chart on the left-hand side shows why glaucoma. Glaucoma actually accounts for almost 12% of the blindness cases worldwide, just behind cataract. Cataract is the most common cause. This is World Health Organization data. I think this is a very old one. I think it's 2013 or something. But I'm sure even now, glaucoma must be a second leading cause of blindness worldwide. So, and next is, what is the epidemiological relevance of glaucoma? Now, as you can see from this graph, that there is an increasing trend in prevalence of glaucoma worldwide. A recent meta-analysis stated that the glaucoma may, among the ages between 40 to 80 years would cross 100 million by 2040. And then what are the risk factors of glaucoma? What are the known risk factors? Aging, ethnicity, gender, then systemic diseases like diabetes, hypertension, clinical factors like elevated IOP, corneal thickness, myopia, and of course then there is family history, which takes care of the genetic factors. So overall, if you see, glaucoma is highly multifactorial with a very complex etiology. The age-dependent increase is highest among Hispanics and whites as compared to Africans and Asians. It was also seen that men had 32% higher risk of POAG, and with no evidence that gender across differed. Men showed higher risk of glaucoma across all the ethnicities. So that slide actually highlighted that both race, ethnicity, and gender was a risk factor for glaucoma. Now, straight away moving to glaucoma genetics, there is abundant evidence today to suggest that genetic factors play a role in glaucoma development in both familial and polygenic types like primary, open angle, angle closure, and pseudoexfoliation. This is just a long list of the genes that are involved with different types of glaucoma, involving familial and non-familial types. And this is just a small list, but there are over 100 genes that have been associated with different types of glaucoma. And investigation to search for more genetic loci which are associated with different types of glaucoma is still an active area of investigation. So when we talk about genetics, so what is the heritability of glaucoma? Heritability, which is represented by H square, is a more complex statistical measurement ranging from 0 to 1 that describe what percentage of variability in a trait is due to genetic factors versus environmental factors. The trait caused by single gene mutations have heritability which is close to 1, indicating that the variability in this trait is entirely genetic. Then studies, when they studied about glaucoma, the heritability estimates in POAG was found to be in the range of 0.17 to 0.81 and 0.65 for angle closure glaucoma, and I think it's close to 0.5 for pseudoexfoliation glaucoma. So as you can see here, since glaucoma is a complex multifactorial trait with quantifiable genetic influence, the H square estimates the heritability falls between 0 to 1, indicating that there are other factors besides genetic. So thus, beside aging, gender, race, and genetics, other environmental factors such as geographical location, exercise, systemic disease also known to contribute to glaucoma. If you can see in this, just to figure out on the right hand side, we can see there have been environmental factors. Like you can see an example with the people staying in northern latitudes are more prone to exfoliation syndrome than women with on postmenopausal hormone therapy are more risk for POAG. And so you can see there seem to be different factors which can also contribute to the risk of glaucoma, which indicate the role for epigenetic regulations. And epigenetic together with gene-environment interaction can influence disease development and progression. So what are these epigenetic regulations? This is just a cartoon to show the central dogma of genetics, where DNA is converted to RNA and RNA is then translated to proteins. Now these processes of transcription and translation, they are influenced by epigenetic regulations. Some of these epigenetic regulations are DNA methylation, then you have chromatin remodeling or histone modification, and then non-coding RNAs. In contrast to genetic mechanisms, epigenetic changes can refer to modifications of the DNA molecule or associated protein that alter gene expression without actually changing the underlying DNA sequence. And epigenetic mechanisms play a critical role in the pathogenesis of complex polygenic human diseases. And today we will focus on non-coding RNA, which is a microRNA. So this is where actually microRNA comes into picture, which is an epigenetic regulator. So what are microRNAs? MicroRNAs are short single-stranded non-coding RNA molecules. They range around 20 to 24 nucleotide in length. They bind to mRNA in the 3' untranslated region of a gene and regulate gene expression by promoting either translational repression or degradation of the mRNA. And as per the MIR-based database, there are almost close to 40,000 entries of different types of microRNA. Now, microRNA genes are located throughout the genomes and introns and rarely in the exons. Importantly, they regulate 30% of the coding genes, and also a single microRNA can target multiple microRNAs. And these microRNA take part in diverse biological function, including cell differentiation, proliferation, apoptosis, development, immune activity, immune response, including in the ocular cells. This is just a small cartoon. We'll see how this microRNA is actually synthesized, the biogenesis of microRNA. So this is inside a cell. In the nucleus, the gene is transcribed to produce primary microRNA, which is then acted upon by a critical enzyme called DROSHA to form a precursor microRNA. The precursor microRNA is then transported into the cytoplasm by exportin, which is again acted upon by dicer. It's again a second critical enzyme, which to form a microRNA duplex. Then these microRNA duplex are converted to single strand, where they can they combine with this RNA-inducing serine complex, where they can act upon the target gene and either have form a translational block or degrade the mRNA target. And the single strand can also be transported back into the circulation via exosomes or aragonate proteins or HDL molecules. So this is just the cycle for microRNA. This cartoon shows now how these different types of microRNA look like. So the entire microRNA structure is actually representing the primary microRNA, which is cleaved by DROSHA to form a precursor microRNA, which is acted upon by dicer to form the mature microRNA. So this is a 5' to 3'. So the 5' sequence is read as microRNA with a 5p, and the mature microRNA, which is in the 3' is labeled as 3p. This is just to tell you how the microRNA are labeled. And the main region where the microRNA binds to the 3' translated region is called as the seed region, and this is the stem region or the loop region. As I said, that a single microRNA can bind multiple gene targets. This is just an example to show how MIR29B can bind to 3 different genes. Here it is BMP1, ADAM12 and NKI-RAS2. What also you can notice here is that the MIR29B is showing a perfect binding over here. But even if it is an imperfect binding, microRNAs can bind to the target gene. So here you can see this is again an example of imperfect binding, and here you can see even though it's not a perfect complementarity, the microRNA can still bind and exhibit their functions. So single microRNA can target multiple genes, and also they can have both imperfect and perfect binding to regulate gene expression. So why are these microRNA important? What is the importance of the microRNA? MicroRNA has been studied to actually form the basis of biomarkers. MicroRNA are readily detectable and differentially expressed in multiple body fluids. What this slide shows here is that these microRNA can be readily detected in different body fluids, and this is just an example where this is detected in 12 different body fluids. I'm sorry, you may not be able to see these things, but this is plasma, urine, CSF, or tear, all these different body fluids where microRNA have been detected, and it is showing a differential expression as well. So that's why there is a lot of interest in this. Also, it has been noticed that even in adverse conditions, maybe high or low pH, boiling, freeze-thaw cycle, the microRNA are quite stable. And this is just to show how microRNA can be analyzed. You can derive microRNA from different body fluids, analyze and isolate and quantify. So these microRNA basically feature as an ideal biomarker, which can be non-specific, and you can be specifically quantifiable, and you can be robust, translatable, predictive, and sensitive. So it just serves as an ideal biomarker because it fulfills all these features that is required for biomarkers, that you can get it from samples very easily. And thus, besides their regulatory role in different human diseases, microRNA also represent promising diagnostic biomarker for clinical use. That's why microRNA is very important. So now this is just an example to when you search PubMed with microRNA and glaucoma, you can see that the publications are increasing. There is an increasing trend in publications since 2009, and there are only 172 publications. This is just with using the term microRNA and glaucoma. So it's quite in infancy, but there is a rise in the interest in this microRNA and glaucoma investigations. So the question arises if microRNA are present in ocular fluids and tissues. Yes, they are present in aqueous humor, in serum, in tear, and these are the different tissues like retina, RPE, cornea, and lens. So what this slide shows is that microRNA are expressed in ocular fluids in different tissues related to the eye, and in glaucoma, the expression of microRNA has been shown to be greatly altered in the cornea, ciliary body, TM cells, and the retina. As you can see from this heat map over here, there is differential expression, like the color coding is different. If you can see, this is like from minimum to maximum microRNA. So yes, they are present in ocular fluids, and they do show a differential expression in different types of tissue and samples. What is not known yet is whether these altered expressions are key cause of development and progression, or mere reflection of physiological changes, which is actually an active area of investigation. Now we'll look at the functional roles of specific microRNA relevant to glaucoma. Now the trabeculum meshwork. As everyone knows, the TM is a critical structure responsible for draining aqueous humor from the eye and producing extracellular matrix. And in glaucoma pathogenesis, primary factor responsible for IOP is impairment in aqueous humor outflow through the trabeculum meshwork. Oxidative stress has been implicated in both initiation and progression of pathophysiological phenomenon in TM dysfunction in glaucoma. And MIR29B was reported to regulate multiple genes in the extracellular matrix protein in the heart cells. So using this hypothesis, these investigators, Luna and colleagues, they investigated the role of microRNA29B in the regulation of ECM in human trabecular meshwork cells. So what they did was, they used primary human TM cells and incubated them in chronic oxidative stress, and the gene expression was evaluated using microarray and confirmed by quantitative RT-PCR. What they noticed was chronic oxidative stress induced down regulation of microRNA29B in the human TM cells, and this decrease was associated with increased expression of several ECM genes, including collagen, LAMC1, fibronectin, and several other genes which are involved in ECM deposition and remodeling, and that's known to be supposed to be regulated by MIR29B. And what they saw was that the increase in expression of all these genes was inhibited when the human TM cells were transfected with microRNA29B mimics. So coming to the first part, they noticed that microRNA29B under chronic oxidative stress was downregulated. So these are the three primary cell lines that they used, and they observed that the levels of microRNA29B was downregulated. Then next part, what they saw was when the human cell lines, the TM cell lines, were transfected with microRNA29B, all these genes which were upregulated when the 29B was downregulated were all inhibited. You can see this is a negative slope line, and these are actually it's a fold changes. In all these are the color coding is for different cell lines compared to array. So what they noticed was all these genes when transfected with MIR29B were downregulated. So MicroRNA 29B was demonstrated to negatively regulate the expression of multiple genes that were involved in ECM in the TM cells. And downregulation of MicroRNA 29B might contribute to several ECM genes under chronic oxidative stress and increase the glaucoma risk. They also observed a similar effect when the cells were treated with, besides oxidative stress, they treated the cells with TGF-beta, and the cytokines also behaved in the same way. They observed the same effects, that MicroRNA 29B was downregulated and all these genes were overexpressed. So they concluded that MicroRNA 29B was an important modulator of ECM synthesis in the human TM cells. Now this is another microRNA, MicroRNA 204, which was found to regulate pathway in the tubercular mesh. Now FOXC1 is a transcription factor that affects eye development and also cause for Axenfeld-Rieger syndrome, which is often accompanied by glaucoma. And FOXC1 is an important protein in TM cells that resist oxidative stress and maintain cell homeostasis. One study found that MicroRNA 204 reduced the expression of this FOXC1. And MicroRNA 204 also downregulated the expression of clock, PLKH5, G5 gene, IGTB1 and MES2 genes in the human TM. So along with this thing, the 204 also downregulated the expression of these genes. So what are these genes? Now these genes, MES2 was a transcription factor which is very essential for development. Clock is a regulates circadian rhythm. Plextrin homology is important for cell signaling and angiogenesis. IGTB plays an important role in immune response. So what the authors concluded was this: the FOXC1 targets these genes in the TM, suggesting that the gene expression in TM is regulated by a complex regulatory network of transcription factors such as FOXC1 and MERIT204 might be involved in this regulation. So this study showed that MicroRNA 204 can also regulate TM homeostasis via regulation of certain important genes. Now regulation of TM contraction and IOP by, there is this Now regulation of TM contraction and IOP by MERIT200C. Contraction of TM cells can result in a decline in the permeability of TM and affect discharge of the aqueous humor. Inhibition of contractile machinery in the TM is an effective method to lower IOP, and MERIT200 family was found to be a potential regulator of ectomyosin system in the TM cells. This study demonstrated that MERIT200C is highly expressed in human TM cells and also inhibited important genes that were involved in actin regulation and related to TM contractions. So you can see that the MERIT200C was highly expressed in TM cells, and when these cell lines, this human TM cell lines were transfected with MERIT200C, these genes were downregulated. This is a control which was transfected by scrambled, and this is this one is when it was transfected with MERIT200C. So like that you have control and 200C transfected. So you can see all these three genes are downregulated when transfected with MERIT200C. They observed that when the cells are transfected with 200C, it exhibited significant decrease in the contractility in response to mediators using a collagen contraction assay. So this is a collagen gel contraction assay. When the cell lines were transfected with control or a scrambled nucleotide, you can see there is contraction of collagen. But when they were transfected with MERIT200C, this gel contraction was inhibited. And this is the fold changes. You can see the gel contraction was decreased in cells which are transfected with 200C. Also they observed was that the transfected cells showed a reduced TM cell traction force when they stimulated with serum on confocal microscopy. This is another experiment where cells are transfected with fluorescein dye, and then you see how the cells move before and after treatment. So you can see these are the blue dots resemble pre, and the purple ones are the post one after the treatment. So what they observed was the TM cell transaction force was reduced in the cell lines that were treated with MERIT200. So this experiment showed them that MicroRNA 200C inhibited TM contraction and reduced cell traction force. They also studied modulation of MIR200C in vivo for IOP. What they did was they injected MIR200C in the anterior chamber of rat eyes, which resulted in decrease in almost 25% decrease in IOP. This is the raw data kind of thing. This is a fold conversion. So let's focus on this one. So here you can see they did actually two injections, one on day 2 and day 5. Well, after first injection they saw that the IOP reduced. As they said, it was almost 25% decrease in IOP. And on second, so this again it normalized. If you see, it normalized after a few days, I think the 6th day. And then they did a second injection and that sustained for a little longer time. The follow-up of this study was around for 17 days, but it was a kind of a proof of concept that in vivo even to MIR200C can modulate IOP. This was the first study that demonstrated the ability to regulate trabecular contraction and modulation of IOP in vivo, making 200C a worthwhile candidate for exploring ways to alter trabecular meshwork contractility, having a therapeutic purpose in glaucoma. Similarly, this was another study which showed IOP regulation by microRNA cluster, MIR143 and 145. So the cluster means that these two microRNA were located on the same gene locus. So these microRNA was found to be expressed abundantly in TM cells and smooth muscle cells in the eye. This is just a relative fold changes where you can see all these cell lines, be it UVEC or human TM cells or smooth muscle cell, they all abundantly express of both these microRNA. And unlike MIR200C, MIR143, 145 was reported to regulate contraction and enhance contractility. Like MIR200C was found to reduce contractility, here it was found to increase contractility and by lengthening F actin and thereby increasing IOP. So it was just the opposite effect of 200C. So what they did was when they did a double knockout of these two genes in mice, it resulted in reduction of IOP. You can see here this is a wild type and here with this double knockout of both these genes, and you can see there was a significant reduction in IOP. So they found that IOP was reduced after double knockout, and also they found that there was a significant increase in the outflow facility in double knockout mice. So here you can see the aqueous outflow is highly significantly increased in the double knockout. So like the IOP was actually decreased because of the increase in outflow facility. The study suggested that these two microRNA are required for maintaining actin cytoskeletal dynamics and contractility in human TM, and manipulating these two microRNAs may reduce IOP and increase outflow, thus having a therapeutic implication in glaucoma. This is another example of MIR21-5P, which was found to regulate IOP by modulating multiple genes. So here what this study did was, they applied these mimics MicroRNA21-5P topically applied to C57BL6 mouse eyes, which and observed significant increase in MIR21 expression in the conventional outflow and reduced IOP. So once, of course, when you apply MicroRNA25P, the expression was increased, and what they found was there was a conventional increase in outflow and reduction in IOP. The pathway analysis showed that there was involvement of multiple genes, P10, ENOS, RHO, MLC, TIMP, and MMP9, which regulated cell adhesion, cytoskeletal, and ECM turnover, were also significantly altered after transfection. The study showed that MIR21P seemed to regulate IOP by modulating multiple genes and might be representing a new therapeutic option in glaucoma. Interestingly, there was another study that used a nanoparticle mediated delivery system of MIR21-5P in mouse model of glaucoma, and they reported a similar effect. They found that the IOP was reduced and aqueous outflow pathway was also increased, and they reported that it was partially through ENOS pathway, supporting the role of MicroRNA21P by using this nanoparticle delivery as a promising tool for anti-glaucoma treatment. Then this is another study that reported MicroRNA182 that inhibited RGC, retinal ganglion cell apoptosis. Now this reagent, which is tetramethyl pyrazine, possesses the therapeutic potential via mechanisms mediated by antioxidation, anti-apoptosis, and is also neuroprotective. What they saw was primary RGC treated with H2O2 undergo apoptosis, which is inhibited by this drug TMP. So primary RGC cells, when they were treated with H2O2, This actually is showing when the cell lines were treated with increasing concentration of H2O2, there is decrease in viability, which is increase in apoptosis. The apoptosis increases, therefore the viability goes down. But this apoptosis inhibited when the cell lines were treated with TMP. Now you can see on this side, when the TMP concentration increases, you can see the viability is increasing. So this reagent was found to inhibit apoptosis. What they found was this effect was mediated by increased expression of microRNA 182. So like you can see in this one, the cells that are treated with TMP, the expression of microRNA 122 was significantly increased. And other way around, knockdown of MIR182 resulted in loss of this protective effect of TMP. So what you can see in this graph, the bar graph on the right hand side, is when you treat with a microRNA inhibitor, like it's present in the last one, so you can see the apoptosis increases. So that inhibition effect of TMP is lost. So they concluded that microRNA 182 inhibits RGC apoptosis. Now this is another study that reported effects of MIR100 and MIR96 in RGC apoptosis. MIR100 levels were reported to be altered in retina, and these cells, the microRNA levels were investigated in RGC, retinal ganglion cell, a cell line treated with H2O2 to induce oxidative stress. What they found was oxidative stress upregulated the expression of MIR100 and induced apoptosis in a dose-dependent manner. So you can see when oxidative stress is induced in the presence of H2O2, the levels of microRNA 100 increases, and also the apoptosis is also increasing. On the other way around, when they found like when this MIR100 is downregulated, it protects against the oxidative stress and induced apoptosis and permitted nerve growth through this phosphorylation of the AKT and ERK pathways. So you can see here microRNA 200 downregulation reduced apoptosis. So when in the presence of inhibitor, apoptosis is decreased, and from 55 to around 30%. Also, when downregulated, it promoted nerve growth. The neural length is increased in these RGC5 cell lines. The data revealed the functional role of microRNA 100 in retina and suggested that it could be associated with glaucoma pathogenesis in which RGC cells are preferentially damaged. Now this is another microRNA 96, where a study by Wang et al. Reported upregulation of MIR96 significantly reduced red cell viability and induced apoptosis. So here there is upregulation of MIR96 and again reduction of cell viability, that is there is induction of apoptosis, and this was found through activation of caspases, namely caspase 2. Caspase 2 was found to be a direct target of MIR96, and they observed that silencing of caspase 2 rescued apoptosis in RGC5 cell lines induced by upregulation of MIR96. MIR96 was reported to be an important regulator of RGC5 cell survival and apoptosis, possibly via caspase 2. Now before we look into the differential expression studies of these microRNA, now these were the studies that actually showed some functional effects, how microRNA could be involved in different pathogenesis of glaucoma. But there are also studies which have reported differential expression of these microRNA in different types of glaucoma. So before we move on to that, we'll just have a quick look at the different types of microRNA profiling tools that are available, how they are actually done. So this first one is by, you can quantify microRNA using real-time RT-PCR. So this method is called as stem loop. So here the microRNA are amplified, converted to cDNA. Via stem loop primer, you design a primer in a specific manner and then go on amplifying them, have a cDNA synthesis, and then, so that this process is called real-time RT-PCR. The only interesting is they design a stem loop primer as compared to normal where you design your single strand primers. That's the difference over here. Then the second method is RNA sequencing. Now for this method, isolated microRNA are ligated to adapters, reverse transcript with an RT primer linked to unique barcode, because this uses high throughput sequencing methods. So they are ligated, the adapters are ligated, reverse transcribed, then you have RT-PCR using unique barcode, convert them to cDNA, and then you go through a high throughput sequencing. This procedure is almost similar to rest of the NGS procedures, where you align and do a quality control, and then finally do your differential expression. So this is also a common technique which is now used for differential RNA expression of RNA, microRNAs. Another technique which is available is the microarray platform. Again here, this involves isolation of RNA, and then you have these probes which are labeled, which are actually attached onto a microarray, and they are having fluorescent labels. What the complementary microRNA go and bind, and these signal intensities are measured and quantified. So this is another procedure using microarray. Now I spoke a lot about how they find targets for microRNA. Then microRNA 96 will target a particular gene and things like that. So how do you do that is by using this reporter assay. As I mentioned that the microRNAs normally target the 3' UTR regions of a target gene. So what you do is you clone this target gene into a luciferase reporter vector. This is a luciferase, which is a fluorescence vector. You clone the gene 3' UTR into the vector, and then you transfect, do a co-transfection of both of these into a cell. What happens is, like as I said, when a microRNA binds to a target gene, it will inhibit translation and it will degrade the RNA. So if the microRNA is binding that gene, then what will happen is, when it binds, there will be translation. You will find that the light intensity is reduced. So that means that the microRNA will bind the target. When the microRNA does not bind your target, then you will just have the emission of the light and you will see a fluorescence. When you convert that to fold chain, you will see something like this. If the target doesn't bind, then you will see a light intensity which is much higher, and if it binds, then it is decreased. So this is basically called a reporter assay, which is commonly used to do a functional studies of microRNA and their target genes. Now we'll move on to this differential expression studies in microRNA and glaucoma. So this first study is about differential expression in microRNA in the aqueous humor of patients with exfoliation glaucoma and primary open angle glaucoma. So this study investigated microRNA expression in aqueous humor samples from 12 POEG and 12 pseudo-exfoliation patients using microRNA expression array. So as I mentioned, now you know what is array and all everything. That's why we just introduced you to the profiling tools so that you know what was done exactly. So they used an array system. So what they found here was 3 microRNAs were significantly different in POEG as compared to controls. They reported 5 microRNA that were differentially expressed in the pseudo-exfoliation as compared to controls. And they found 1 microRNA which was common to both XFG and POEG, both exfoliation and open angle glaucoma. So what they concluded was that the microRNA profile is distinct between 2 glaucomas with one overlapping, although there was an overlapping one, but it can actually distinguish between 2 types of glaucoma, suggesting that the microRNA profile can discriminate between glaucoma types. The pathway analysis showed them that these microRNA were involved in potential glaucoma pathways, including focal adhesion, tight junctions, and TGF-beta signaling. Importantly, they noticed that this microRNA 122.5p was also could target OPT and optineurin, TGF-beta1 and TMCO1, which are actually known glaucoma genes. Now, circulating microRNAs. Now, considering the involvement of vascular dysfunction in glaucoma, which is more common to POEG and exfoliation, there are studies that investigated circulating microRNA in peripheral blood or plasma rather than the eye. Like earlier study reported in aqueous humor, but there are studies which have investigated in peripheral blood and plasma as well. And peripheral blood and plasma are readily accessible samples that can be easily assayed in clinical labs and would greatly facilitate identification of blood-based biomarkers for this disease in the future. So coming to the first study, this study reports differential expression in peripheral blood in pseudo-exfoliation. So this study reported overexpression of 12 microRNAs from peripheral blood, though the sample size are small. All these studies were, because as I said, these are in their infancy. So they actually provide a proof of concept rather than no large studies have been done. So they studied 17 exfoliation glaucoma, and then patients with ocular hypertension and 11 controls, and they reported this differential expression of these 12 microRNA. Again, when they looked at the pathway analysis, they found that these microRNA targeted important genes that were involved in ECM extracellular matrix of homeostasis of glaucoma pathogenesis. Now this is another study which compared both plasma and aqueous humor samples into ethnic cohort from glaucoma. This is again a mixed population of patients, POEG, pseudo-exfoliation and syndrome. and plus compared to controls. Now here they used qPCR. So what they observed first thing from both this plasma and aqueous humor that both of them reported high levels of microRNA. The number of detectable microRNAs are much higher in both aqueous humor and plasma samples compared to cataract. Now, sorry, this is a very busy slide, but what it is showing is they actually observed 20 plasma microRNA were having at least 1.5-fold higher expression in glaucoma and both pseudoexfoliation across the two ethnic cohort. What they studied was some patients from New York ethnicity and other from Japan. So these are the two ethnicity groups that they studied. And consistently across both these two ethnicities, these 20 microRNAs were differentially expressed, significantly differentially expressed in both XFG and POEG patients. Interestingly, of these, 6 microRNA were significantly increased in AH as well, of the same subjects. So these 6 microRNA actually were overlapping between plasma and aqueous humor. And they also observed was a combination of plasma microRNAs. Let's see, this 1, 2 and 3, combination of 3 microRNAs allowed discrimination between glaucoma patients and control subjects, and the area under curve was found to be 0.91, which is actually AUC is an indicator of the diagnostic potential, and 0.91 was found to be a strong indicator of diagnostic potential of these combinations of microRNA to actually diagnose the glaucoma cases. So they reported that screening of a combination of microRNA, instead of looking for one single microRNA, screening for combination of microRNA would have more diagnostic relevance. Coming to the possible mechanisms, they found that these microRNAs induce neuroinflammation, involved in eNOS signaling and neurotropin receptor signaling. Neurotropin, as you might be aware, is implicated in RGC survival both in animal models and in humans, suggesting this potential involvement of these microRNA in glaucoma. And relevance of nitric oxide is also very well established. Genetic association studies have already reported multiple polymorphisms to be associated with glaucoma, and nitric oxide is a novel target for lowering IOP. So these findings provided further support of the importance of microRNA in glaucoma pathogenesis and their potential utility as biomarkers. Now, there are also studies that reported microRNA profiling in tear samples. This study reported microRNA signature from tear in patients with POAG and ocular hypertension, and they used was next generation sequencing. They identified 8 microRNAs that were differentially expressed between POAG and ocular hypertension. This is the first study to identify microRNA in tears from individuals with OHT and glaucoma patients, revealing its potential molecular diagnostic biomarker in glaucoma suspect. So what these studies are showing, like these samples can be utilized to identify potential biomarkers, potential microRNAs that can be used as biomarkers. Again, from all these 8 microRNAs, these are the different AUC what they observed, and what they saw was the 4 microRNAs were having a AUC of greater than 0.75, which is again a good indicator for predictors of POAG. Oops, sorry. And the target genes were all found to be relevant, which are involved, like IOP regulation, extracellular. This is just a pathway analysis what they performed to see where exactly this microRNA can function. So these are all the areas which are all relevant to glaucoma function. So these microRNA targeted genes and signaling pathways related to aqueous humor homeostasis, extracellular matrix function, oxidative stress, apoptosis, inflammation, neurodegeneration, highlighting the importance of these microRNA in glaucoma. Now, lastly, I think this is for profiling of microRNA in aqueous humor.
We need to get the timing.
Oh, okay. We are done?
We need to have 5 minutes.
Oh, okay. I think we are nearing. Almost it's getting over now. So again, this is just, we'll go quickly through this now. Now this reports aqueous humor and normal tension glaucoma using RNA sequencing. Again, differential expression of certain RNAs, microRNAs have been reported, and all these are relevant to important pathways which are involved in glaucoma. Now, what is the link between microRNA and glaucoma genes? So genetic variants in microRNA associated with glaucoma and phenotypes. So what we spoke about right now was functions in glaucoma, microRNA differential expressions, and there is also polymorphisms that are reported in these genes which are relevant to glaucoma. So this is a common variant that was reported in MIR182, which we saw that it was involved in RGC apoptosis. Then there was another RNA variant which was reported in microRNA 612 and another microRNA, and both these microRNA were found to be associated with levels, expression of these microRNA, which can actually affect the downstream pathways, and these microRNA was also involved with one of the target genes which is involved in glaucoma. There is a report of another NRIL, which is called as a long non-coding RNA, which is transcribed in the anti-size direction, which is also called as a CDK2 and 2B and 2A gene cluster at 90P. So polymorphism has been also reported in this gene, and the mechanism is thought to be more of an epigenetic rather than genetic, which is because it is involved in cell cycle regulation, and it is hypothesized to affect RGC apoptosis and optic nerve vulnerability. We also reported polymorphisms in dicer and DROSHA. As I mentioned in the biogenesis, there are two critical enzymes which actually control microRNA biogenesis. So we saw there was a polymorphism in the dicer 3' UTR, which was protective in exfoliation glaucoma. We found an haplotype also with both these enzymes, which was associated with protective effect in glaucoma. And what we hypothesized was this polymorphism will affect dicer levels, or they can affect microRNA levels, which might affect the downstream processes involved in TM homeostasis and thereby affecting a glaucoma outcome. So this is just quickly to show that there was a microRNA 124P which is involved in TGF-beta regulation, which is a known pathway implicated in pseudoexfoliation glaucoma, and that this microRNA was actually a target for dicer 1. Supposedly, predicted to be a target of dicer 1, and enhances binding and reduces the expression of this microRNA. And when the expression of this microRNA will be reduced, this pathway will also be downregulated and thereby exerting the protective effect. This is another polymorphism in the ACVR1, which was present in 3' UTR region, which was associated with glaucoma. This gene is a Wnt signal antagonist. That means it inhibits Wnt signaling via these Wnt inhibitors, and this Wnt signaling is very important for maintenance of TM homeostasis and maintaining normal IOP. We found that these two microRNAs were target for these genes, and because of the variant, this regulation may be lost, which might result in an increase in Wnt inhibitors, which will inhibit Wnt signaling and thereby increasing the risk of glaucoma. Finally, to conclude, we saw microRNA can modulate TM function, which is a major structure responsible for controlling IOP. They can regulate IOP, which is an important endophenotype for glaucoma diagnosis. They can affect RGC survival and reduce apoptosis of RGCs. This can indirectly increase the quality of life of patients with glaucoma. And microRNA profiling studies showed that they are differentially expressed in glaucoma and glaucoma types, even in tissues, aqueous humor, plasma, tear, highlighting their potential utility as important biomarkers to aid diagnosis and monitoring of progression of glaucoma. A combination of several microRNAs with existing clinical diagnostic method may help in early detection rate in the future. These studies suggest that microRNA have potential to be used as biomarkers, but they require further research in larger cohort and develop standardized protocol for comparison of studies and analysis of microRNA in clinical samples. Then in addition, microRNA appears to be highly well suited for local transient therapeutic applications, as we saw that microRNAs could regulate IOP, so they have therapeutic potential as well. But the complete understanding of their function, effect of microRNA manipulation, clinical validations, and challenges to delivery are yet to be explored. These are just a few examples. Whatever we discussed, a few examples, a few studies that were conducting on the role of microRNA glaucoma. There is still a lot more to learn on microRNA, different types of glaucoma, but the emerging evidence do suggest that they have a potential for biomarkers and therapy. And also considering the complexity of polygenic and multifactorial nature of glaucoma, an integrated approach of using both genetic and epigenetic studies would be very helpful to eventually develop gene-based therapy or microRNA-based therapies. Thank you.